Okay, so I'm going to put recording on here. Just starting today's lecture, I want to show you real quick where this is, where our lecture slides will be. If you go to Content in Blackboard, there is a YouTube playlist here for all the lecture recordings. And you can see here that the first lecture from last week is up there already. I'll post the other lectures as we go. YouTube is supposed to have a 10-minute limit per video, but for some reason in my account they allow me to upload longer videos, a few hours. So as long as YouTube allows me to do this, we'll record the videos this way. Otherwise, I'll upload the files to Blackboard, and it's less convenient. YouTube is more convenient this way. So I'm going to try keeping this updated after class at night. I'll upload the lecture notes. The most delay you can expect to get is probably like a few days. Once in a while we may have some kind of technical issue where the lecture recording didn't work for some reason, so bear with me here since the recordings, the way we do the recordings is not perfect. So far it's worked really well, so let's just stick with that. So then let's continue. Like I said, we're just taking a snapshot of different examples of BioMEMS to just illustrate some of the benefits. I want to bring up this slide again so you can just touch on these. There is, among the different benefits of BioMEMS, there's a small scale benefit, exploiting microscale physics, the fact that devices can be the same size as cells and biomolecules, portability of different portability of the devices, reducing the reagent costs. Then there's the high throughput benefit. The fact that your devices are smaller allows you to put more assays or more devices in a given area, so you can do parallelization. You can use batch fabrication techniques to reduce cost, and you can do complex fluidic control, as we saw with Quake's work. Then there's a quantitative benefit. Quantitative benefit means that some of these systems can give you a quantitative answer. Okay, a few examples of that. When you can integrate multiple functions on a chip. Let's say you want to do some kind of blood analysis, you do some kind of sample preparation at the beginning, then you do the separation of cells from the serum, then you do analysis of the serum, and then you have the detectors on chip also to do the actual analysis. So that can give you a quantitative result by having the integration of multiple functions on chip. Another advantage to quantitative benefits is what was just mentioned. What was your name? Remind me? Nick. Nick mentioned laminar flows earlier today. So laminar flows are very predictable. Right? So when you have predictable flows, you can have predictable diffusion times, predictable shear rates on whatever's in the microfluidic channel. So that allows you to have predictable transport, predictable quantitative results that way. Microfabrication technologies allow us to make channels with very predictable dimensions, with small tolerances. So there's a quantitative benefit to the precision that you get when you make some of these devices with precise manufacturing techniques. Another aspect of it, which we haven't talked about too much yet, but in a lot of statistical assays, actually we'll start with this, a large number, repeating the same assay multiple times actually allows you to get statistical results on a population. So let's start with that today. That's where we left off. So one of the areas that biologists are very interested in is this area of high throughput single cell studies. So with traditional biology, we would study a large population of cells in a petri dish. Okay, so we played out a bunch of cells on a dish, and what we're doing there is we're looking at the response of the cells in a population. Okay, so if I were to draw, by the way, I've tried this with my other class, and it worked pretty well. So I have started using OneNote as sort of a digital notebook. So let's. I was planning on doing that for this class as well. So let's try that out. Yeah, this way, some of my students always said, well, your lectures are recorded, but you can never record what's on the chalkboard. So I'm going to draw some stuff out here for you all. So for those of you who aren't familiar with this, if I can just, this is a very crude drawing. But you have a plastic dish like this and a cell culture. You might plate all your cells out at the bottom here like this. Okay? And then, for example, if you're trying to do some type of measurement on the cells, what's most commonly done is that you have a microscope here and you just physically observe the cells. Okay? Now, this is a typical cell culture. All right. So one of the things you notice about this is that all the cells here, they're all in the same solution, right? And furthermore, they're all next to each other. So there's actually a lot of cell-to-cell communication that happens, especially in mammalian cells. Furthermore, whatever one cell is secreting into the microenvironment here, like this, is being picked up by all the rest of the cells in that microenvironment, right? So what you're doing is when you measure something, whether you're measuring it optically, this is an optical measurement. Another type of measurement which can be done is if you do a direct pipette measurement of what is being secreted by the cells, how the solution changes. For example, when cells uptake nutrients from the solution, or when they excrete waste products. Sometimes the cells are secreting proteins or antibodies. Okay, those can be measured by taking a pipette and taking out a little bit of that solution. But when you're doing that, you're actually measuring the ensemble properties of the entire population, right? You're averaging out the response of the entire population. So then we get a situation like what we have here. There you go. So this is what we refer to as a cell ensemble analysis. Okay, so you have a petri dish, and these cells might behave slightly differently. There might be cell-to-cell differences. However, what we're measuring is just a single response for the entire population, right? This graph down here shows an example of one type of measurement. Let's say we're culturing a whole bunch of bacteria in a dish. Okay, the bacteria multiply, and so what happens is that the turbidity, the optical density of the solution literally gets darker, right? So the turbidity of the solution over time goes something like this as the cells grow, as they multiply. All right, but what's happening is that you're getting a measurement of the entire population as a whole. This is what's called an ensemble measurement. Okay, something very interesting that you can do with microfluidics is this idea of single cell assays where each cell is in an isolated container. All right, so going back to this idea, if instead of this idea, if we could have a cell in a container, a cell in a container, a cell in a container, and so on, now we don't have cell-to-cell communication anymore. Cells are isolated from one another. Another interesting thing here is that if a cell, so let's say this is a chamber, and it's a microchamber. And the chamber is not much larger than the cell itself. Okay? And this little circle in the middle here is the cell. So now let's say this cell secretes something to its microenvironment. This container around it, it's so small that when this secretes, let's say it secretes a protein molecule like this. Okay? So this protein molecule, if this container is very, very tiny, then it's still at a sufficient concentration where it can actually be detected. Compare that to the situation here. If a cell were to secrete a protein of some sort. Finding this protein is very difficult, right? It's like a needle in a haystack because you basically secreted a protein molecule into an ocean. But when you take the cell and you put it into a very small container like this, now if there's a few protein molecules that are secreted by the cell, they can be detected because the size of the container is very small. The concentration is sufficient. Whenever we detect things, we always talk about things in terms of concentration. If the concentration is sufficiently high, we can detect it. All right, so certain times by doing single cell assays like this in small chambers, the effective concentration is increased. You can detect secretions from individual cells like that. Now going back to this, so we can do single cell analysis where we put cells in these small containers. Secretions from the cells are measured by looking at the concentration. Of the whatever's in that container. All right, the containers have to be sealed, so the chambers have to be quite small. Now, how you can implement that is that you can take the cells and you can put them in a microfabricated device that has a whole bunch of wells in it. This is called microwell array. Okay, and the size of the microwells can be so small that in fact they can statistically they can trap individual cells. Where you have this thing called Poisson statistics, where there's some probability that a well will have one cell. There's some probability that you'll have an empty well, like you see here. There's even some probability that you'll have more than one cell in a well, like you see up here. Okay, and that probability can actually be adjusted. Okay. When you do experiments like this, since we can put so many of those microwells into a microfabricated device, you're actually doing tens of thousands of experiments at once. You can accrue statistical data over the entire cell population while studying each individual cell at the same time. So you can imagine if you're trying to look at differences within the cell population, this can be very powerful. And this is something that's just starting to be done, meaning like it's really starting to take off in the last, I would say the last six years or so. This is an example of a work by Luke Lee's group at UC Berkeley, one of the earlier works, where you could trap cells using microfluidics. All right. Now I want to point out that in this particular example, the cells are not in isolated containers, but they are trapped in these little areas like this. How it works is that the cell, you have an array of traps like this, okay, little structures, obstacles in the flow. And then you have a general flow pattern going from top to bottom. Okay, so when the cell approaches this obstacle, it actually falls right into this trap here. The interesting thing that happens due to laminar flows is that when a cell is trapped, you can see here in part B, can everyone see this? The flow lines are actually altered so that the additional cells actually go around this obstacle. Okay, so what happens is that you end up getting a situation where a single cell is trapped into each one of these obstacles. So this, on the right here, is an array of single cells where all the cells are isolated from one another. No cell-to-cell contact. So this allows some degree of single-cell studies. This type of device that you see here on the right offers an additional degree of isolation. Not only is there no single cell-to-cell contact, but they're also in physically isolated containers, so that whatever the cell secretes into the environment is not going into the next chamber. Question?
Here, it moves around the obstacle and goes into the next chamber. In the hydrodynamic cell traps.
That's correct.
In the microwells, how do they move?
So the microwells, this particular microwell array is kind of interesting. What you do is you take you can take an array of cells. There's a few approaches to doing this. There we go. So you could take I'm going to draw this two-dimensionally. You could have an array of wells like this, and then flow, let's say this is your solution of cells, like this. You could flow them through a microfluidic channel. and these cells would fall into these wells like so. That's one approach. Another approach you can do is by putting an array of cells into just a space like this randomly, and then you take a device that might look something like this. It has an array of wells, like so, and you literally take this and you cap it over the top, so that you encapsulate each one into a well like this. Now, statistically, some of the wells are going to be empty, some of them are going to have more than one cell, and so on, but this type of approach has been done as well.
So does that give control over each and every cell, so you can examine what's the difference between each and every cell?
Yeah, so the question was, does this allow you to examine the differences between cells? Yes, I mean, these microwell arrays would be transparent, so you can observe them optically. There has even been work where you actually put electrical chemical sensors inside each one of those wells, so we can detect the secretions electrochemically. But often what is done is that whatever the cells are secreting, what is secreted out into the microenvironment around it, there's some sort of reagent, chemical reagent, that causes that microenvironment to become fluorescent, so it glows. So you can measure the amount of secretions that way by looking at the degree of fluorescence. So I gave one example here. This was the hydrodynamic cell traps. This was the microwell cell traps. A third example of this is cell encapsulation in droplets. You can actually create these little devices that generate drops, microfluidic drops. I'll show you on the next slide. In fact, maybe I can just move ahead here. You see on the left here, I don't know if I can zoom in. Can I zoom in? There we go. All right. So can everyone see this here? This is a microfluidic junction. What is a junction? It's just two inlets coming together at the same spot. Now what's done here is that you flow in an aqueous solution. So what's coming in from the top here is an aqueous solution. What's coming in from the left here is oil. Oil and water don't mix. So something very interesting that happens at the microscale, that if you have a stream of oil coming in and a stream of water coming in, they will actually, the oil stream will actually break off in a repeatable manner, and it'll form a train of droplets in oil. Okay, and this is an approach that now I would say maybe 30%, 20 to 30% of the papers published in microfluidics use droplets in some form or another. Right, so these droplets can actually be very, very tiny. They can be maybe 10 to 30 microns in size, which is really not much larger than a single cell. So one of the things people have done, or people are very interested in, is encapsulating single cells in these droplets. You have a train of drops where there's cells inside of them. Now, one of the challenges right here is that you still get that Poisson statistics. There's a random encapsulation efficiency. Some drops will have 0 cells, some will have 1, some will have 2, some will have 3. It depends on how dense your cell population is into the stream that's going in at the top here. Right? You can imagine that if you have a very dense stream of cells, then you'll have many cells per droplet. Right? But one of the big problems that people are trying to work on right now is how can you get exactly 1? Because we'd like to do single cell assays. Yes?
But do the cells multiply in those wells?
Yes, in fact they do. They've done bacterial cultures, bacterial cultivation in these types of microreactors.
Okay. What exactly is the purpose of having a single cell in each well when they actually multiply eventually?
There's actually quite a few reasons to do that. If there is a variation, if there's variation among cell types, so we assume that all cells in a population are the same, or monoclonal, what have you. But there are actually small individual differences, and these individual differences can actually be even larger. They can be amplified if the cells are exposed to different things. For example, if cells are exposed to a chemical, sometimes the cells have mutations. This represents what happens with us too when we get exposed to ultraviolet radiation or we eat something that's carcinogenic. Some cells manage to stay healthy because they have repair mechanisms. Other cells have more degree of damage, so they start to go bonkers. And things like cancer are really from this type of single cell origin. One cell goes bonkers because of a mutation, and then it starts to grow out of control. Right? So there's a lot of interest in studying the differences among individual cells in a population. When you grow a population of cells inside a microreactor, cell A, which is, let's say, a wild type, grows up and has a normal growth rate. Cell B that has a mutation in it would multiply much more rapidly. So there is a benefit to being able to culture them in separate containers. Okay. So let me just back up here. We'll come back to this slide in a second. So just to wrap up this slide here, the advantage of doing high throughput studies like this is that you can, is sort of what's schematically illustrated here, is that you can sort of get statistical information over the entire population of cells while studying each cell at one time. All right, there's a lot of interest in this, like high throughput studies. So whenever you hear the word high throughput biology, this is one of the areas that they're referring to. Coming back to droplet microfluidics, we talked about how you can create trains of droplets. Now, one of the reasons why you'd want to do that, let's say you want to do hundreds of different chemical experiments at the same time, or hundreds of chemical experiments very rapidly. One of the things that's very useful for is screening through a whole bunch of reaction conditions. Now, for those of you who aren't chemists, just think about this from a chemist's perspective, from a person like making a recipe, all right? Sometimes you want to try out different amounts of what's in a recipe to get the ideal result, right? So someone who's cooking might experiment with different amounts of sugar and see, like what the ideal amount is, right? Now, this type of thing is done in chemistry. It's called titrations, where you're experimenting with a whole bunch of different concentrations of a certain chemical. It has relevance to things like toxicity screening. Let's say you have cells, and you want to find out how much concentration they can tolerate before they start to die. This is called LD50 in environmental chemistry, the lethal dose. The lethal dose that will kill 50% of the organism, right? So these type of toxicity screens, it's useful if you could do a whole bunch of experiments at slightly different concentrations. Or perhaps you want to have different combinations of potential toxins. Maybe there's a synergistic action of two different toxins and you need to do a permutative screen where you take all concentrations of A, all concentrations of B, and you can imagine like how that scales up permutatively. There's many experiments that you have to do. And these are the types of things that are often done in the drug discovery industry. Someone discovers a drug, right? And you want to see how much of that drug is needed to kill off a certain bacteria, right? Another very good example of this was done with protein crystallization. This example right here. And there we go. So protein crystallization is one of those recipe things, okay? Proteins there are many proteins that are soluble in solution, right? They're soluble in the solution. However, in the field of proteomics, one thing that people are quite interested in is studying the structure of proteins, the crystal structure of proteins. So what you have to do in this case is you have to get the proteins to crystallize, to come out from the solubilized form and precipitate into a crystal. Okay, and this is one of those things that was sort of a black art in the sense that it was literally like experimenting with a whole bunch of titration agents and looking at the right rate at which you can add them and having just the right mix of A, B, and C, titration agents A, B, and C. Okay, at which concentrations do you have to use? Sometimes you'd have to screen through hundreds of them in order to get just the right condition in order to get these crystals to form that you can see here. Those are actually crystals of proteins. You can see they're different colors. Now, once you have those crystals of protein, you can put them in something called an X-ray crystallography tool. You can do X-ray crystallography with them. You put them in something called a synchrotron, which puts X-rays through them. It looks at the diffraction patterns through the crystal, and by doing that, you can actually get those beautiful chemical structures that you've seen that some of you have seen in your chemistry textbooks, where they show the actual structure of the protein, what it looks like. That's X-ray crystallography. One of the reasons why that was very difficult was this step, getting the proteins to crystallize properly. So one of the early works by Rustem Ismaghilov's group back in 2003 and later in 2006, they had several papers on this, is that they use this train of drops to screen through a whole bunch of different reaction conditions. Now, if we zoom in here, you can kind of see here that at the top here, there's 3 inlets. So they actually had 3 titration reagents. Well, the protein solution was coming in here, and I think 2 different titration reagents were coming in on this side. So what they did was they adjusted the flow rate of the titration reagents so that each one of these droplets had a different concentration of the titration reagents. Okay, and just in the matter of minutes, they could generate hundreds of droplets, each with a slightly different combination of these titration reagents. And what they found was that in most of the droplets, crystals did not form, but in some of them, the crystals did in fact form. Right? So screening through hundreds of different conditions, this is one of those examples of cost reduction too, because those titration reagents are actually quite expensive. To be able to systematically screen through all those different conditions, and each one of these reaction volumes is only a nanoliter, 10 to the negative 9 liters, 1,000 times smaller than a typical microplate. You can imagine the economic benefits of that. All right, so this is a good example of what it means to miniaturize and why that's useful, specifically in the life sciences. This was an example, another example of biology being conducted in really small containers. They had a packed emulsion of droplets in oil. Now, each one of these droplets contained cells. These cells had been exposed to a mutagen, so that caused some of the cells to secrete a certain type of enzyme more than the other ones. And they could measure it. Even this small example, you can see there's probably several hundred different compartments there, several hundred cells. And you can see that the cells which are glowing green are the ones where the cells were secreting a certain type of enzyme that they were looking for. And basically the cells that were fluorescing brightly had a lot of these secretions. So that was a cell type that they were looking for. So they could sort out the droplets that had these highly fluorescent signatures, and they could look at those cells and study those cells in order to determine what they could look at the genetic makeup, although that's a little bit more difficult. In this case, what they were doing is they were just trying to search for cells that were secreting more of a certain enzyme. Once they selected a group of cells that was secreting more of this enzyme, they again did another round of mutagenesis. So those optimized cells were again mutated, and some subset of those expressed an even higher amount of this enzyme. So a few stages of that, they were able to create a genetically modified bacteria that secreted this type of enzyme that they were looking for. These type of screens require you to screen millions of cells, a population of millions of cells. Okay, so by scaling them down to droplets, this was a really quite an amazing work. This is a paper that was published in 2009, very highly cited. There's actually a company who's using techniques like this. A few companies, in fact. We'll try to talk about this if we have time later in the semester. Another thing that you can do is sort of outside the realm of biology, but just more in the manufacturing area. So we talked about this idea that you can create a train of droplets. Okay, and those droplets are liquids. But what if you want to make solid particulates? You can actually use the system to do that. Let's start with this one here, or let's start on the right here. So I talked about before how you can, if you combine streams of water and oil in a microfluidic device, you actually create trains of droplets. Now, if that liquid is made up of a monomer, a chemical monomer, that can be UV cross-linked. Cross-linked means there are chemical bonds that form between the chains of the monomer. And the short story is by exposing that to ultraviolet light, you can turn that from a liquid to a solid. Okay, so there were droplets being formed in this area here, but after exposure to ultraviolet light, those liquid droplets became solids. And so many people have used examples of that. You can see here where they actually create micro and nanoparticles using these types of microfluidic devices. Now the cool thing is, I mentioned before that you can change the composition of each one of the droplets. Well, you could create particles with very well-tuned compositions, right? You can also, instead of creating spherical particles can be created using a variety of different techniques. But what if you want to create slightly ellipsoidal particles? You can do that using this type of approach. What if you want to create what's called the Janus particle, where a particle has a certain set of chemical properties on one half of it and another set of chemical properties on the other half? It's the equivalent of Dr. Jekyll and Mr. Hyde. Like one face, or you've seen the Batman movie, like Two-Face. What is it? Harvey Two-Face? Yeah. Anyway, you get the idea. Like the particles have different properties on one side versus the other. Very difficult to create that deterministically at a reasonable cost. Like these types of systems can create these types of Janus particles with very, very low cost, really. You can make complex things too. You can make these things called double emulsions. All right, what you see here is these two. This is a water and oil in water emulsion. So these two drops here that you see here, the blue and the purple, those are water phase. Surrounding that is another bubble that's oil, and surrounding that is water. All right, so you can create these things called double emulsions. You can even create triple, quadruple, quintuple emulsions. These things are really impossible to create with any other way. Right? So this is an example of where microfluidics are being used, that not necessarily for biology, but actually for manufacturing new types of micro and nanoparticles. But as it turns out, like some of these micro and nanoparticles, they have applications in chemistry, but many of them also have very interesting applications in biology for implantable devices, drug delivery, and so on. So, these particles are useful for a wide variety of biological and manufacturing processes. Any questions so far? Good. All right, so moving on. Some more benefits in BioMEMS. We'll talk a little bit about the quantitative benefits right now. I mentioned the integration of multiple functions on a chip. Predictable dimensions, fluid flows. So precise dimensions, precise flows yield precise assay results. And some more about the large numbers of assays. So I'll just give a few highlights here. Instead of going over all the concepts, we're just going to talk about a few well-known works which sort of represents what's possible in the microfluidics field. This slide is called Integrated Sample to Answer Genetic Analysis Systems. The philosophy behind lab on a chip was the idea that you could bring your lab and shrink it down to the size of a chip and to be able to do a lot of diagnostics on site. Right? These were some of the early examples which really showed that this was possible. If we go over here on the left side here, this was one of the early work done by Mark Bern's group at the University of Michigan. They were actually one of the first pioneering groups in the microfluidics field. They published some of their first things in the mid-90s. And the microfluidics field really started to take off in the late 90s and early 2000s. In around mid-2005, they published this paper, which was a genetic analysis system. Now, we're not going to go over all the details of how this works, I'll just want to mention a few different things. Okay, these systems integrate a whole bunch of different chemical functions on the same chip. You can think about it as sort of like a chemical factory. What does it combine? It combines a sample preparation. It combines something called polymerase chain reaction and electrophoresis on a chip. How does this do genetic analysis? So you're going to start off with a sample, okay, chemical sample that has, if we take a cell sample from me, for example, my cells are going to have DNA in it. If I want to analyze that DNA, first I have to isolate and extract the DNA, right? Get rid of everything else. That's called sample prep. It's also referred to as chemical separation. Next, we have to, the amount of DNA that you're going to get from a cell is really small amount, right? So you have to then amplify that. PCR stands for polymerase chain reaction. It's used to amplify DNA. If you have just a few copies of DNA, after every round of PCR that you do, it doubles the amount of DNA that you have. So after 20 rounds of PCR cycling, you'll have 2 to the 20th more DNA that you started off with. Okay. By amplifying DNA, you get more of it, makes it easier to measure it. PCR itself, you could spend a whole class talking about PCR. You could spend a whole term talking about PCR. The person who invented PCR got the Nobel Prize. This is how big PCR is, like a mainstay in all biological forensics, what have you, any type of laboratory. So sample prep, then PCR, then you do electrophoresis. Now, there may be a certain strand of DNA that we're looking for. Go back here. Again, it's, without going into all the details, bear with me here if it seems like this is not detailed enough. But I will tell you at this point, for those of you who aren't biochemists, that DNA, when you get DNA from a sample, it comes in you do a digest of it, and you end up with a whole bunch of strands of different length. DNA looks like these strings. I'm sorry. All right. So what you have to do is then separate these strands of different length. You have to send them into something called an electrophoresis device. The electrophoresis device separates those strands out in time so that the short strands come out first and the longer strands come out later. Okay. If I am interested in seeing how much of a certain gene that I have, a certain type of DNA, okay, they're interested in a certain section of my genome, then they can do electrophoresis and just isolate this particular DNA that they're looking for. Things like paternity tests, things like if you want to look at if you have a genetic defect. For example, I have a minor blood condition called thalassemia. Right? It's a defect in a certain part of my genome. So if they want to do a thalassemia test on me, what they would do is they would take my DNA, they would extract out the gene that's related to thalassemia and do an assay on that to see if it has a certain sequence or if it has a certain defect missing. All right. If you want to do paternity tests, like you see on Jerry Springer. None of you watch Jerry Springer. Brian watches Jerry Springer. I don't. I promise. It's just I heard about it. They look to see a certain sequence of DNA which matches the child. Yes.
Is it just for diagnosis, or do we have control over make a mutation so that the disease doesn't?
So the question is this only for diagnosis, or can we actually change the DNA to actually change the properties of a person, right? Organism. What you're referring to is called gene therapy. Okay, that's sort of a new field. It can be done in certain circumstances, but it's still quite new. Yeah. In a few examples where it's more commonplace, it's like genetically modified organisms. You can make genetically modified plants. They don't actually. They can do things by actually implanting DNA into certain parts of the genome. Genetically modified mice, for example, are used in biology labs. Genetically modified plants are actually created by crossing two plants together that have specific properties that you're looking for. That's another hot button topic these days. GMOs. Not all GMOs are bad, but some of them, well, I won't get into that. I'm getting off topic here. The point here is that there's a lot of the genetic analysis involves these three different steps. Sample prep, PCR, electrophoresis, and also you have to actually detect your DNA as well. So this particular chip does all that. Okay, you have sample inlets here at the bottom, and it goes through a set of sample prep portion of it. There's PCR reagents here. Actually, the test sample comes in through here. So it goes through this chamber where the PCR, the amplification of the DNA is done, and then it comes over to this area of the chamber where the electrophoresis is done. You can see those little shiny green bands. That's actually the DNA, and what each band actually represents a certain length of DNA, and however bright that band is, tells you what type of DNA that you have. Okay, we'll get into the more details of that later. But what you can see is that there is an advantage of putting all those functions on a single chip. Other examples of that we saw included the Quake Chip. We back up to that, where you have multiple valves, multiple chambers integrated on the same chip. So you're integrating functionality into the same area. This allows you to do very complex systems where you have thousands of valves, thousands of different chambers, and complex fluidic control. Okay, moving on. So these chips, the Bern's chip on the left there, they had integrated fluid handling. They had on-chip valves. They had on-chip heaters for doing this polymerase chain reaction because PCR requires you to increase and decrease the temperature at several times. Each round of amplification where the DNA is doubled requires you to increase the temperature and bring down the temperature. This is called thermal cycling. The PCR reaction requires thermal cycling, although some of the newer ones are isothermal. They also include electrodes that create electric fields required for electrophoresis. So electrophoresis, where the DNA is moving from one place to another at different speeds, that requires a voltage source. Creates an electric field. And the electric field propels the DNA in a certain direction. Different sized DNA travels at different speeds. Okay? A lot of different functionality in a small area. So the Bern's chip used this chip for influenza detection, detecting certain strains of the influenza virus. The system that you see here on the right also integrates some of those functionalities. You can see here that you have a PCR mixture here. You have, let's see, you have the area where electrophoresis is happening, and so on. So these are, the workflows are similar. The chips are a little bit different in terms of how much is integrated on the chip and how much is not. But the idea is sort of the same. The detection, this particular chip, James Lander's lab was at the University of Virginia. They do a lot of forensics, and they also do a lot of biodetection. So in this particular case, they were looking at detecting anthrax genetically by using this procedure in less than 30 minutes. Detecting anthrax would normally, you'd have to send it off to a lab, and it would take maybe a day or two for you to get the results back.